Hi, I'm Cécile Martina. I'm Director of Research at ISTEM, and I would like to thank the organizer for giving me the opportunity to present the topic that my group is developing concerning the use of human pluripotent stem cell for neuromuscular disease. And all this work is developed in ISTEM, and I would like to spend a couple of minutes on what is exactly ISTEM. So ISTEM is a French institute that is located in the south suburb of Paris, which has been created in 2005. It's a partnership between French Association of Myopathy and ISTEM. And all the activity developed in ISTEM are dedicated to two free keywords: human pluripotent stem cell, monogenic disease, and therapeutics. In other words, our goal is to use human pluripotent stem cell to develop new therapeutic strategy towards monogenic disease. And we have two main axes of research in ISTEM, one focusing on the cell therapy, and the other one is to use human pluripotent stem cell for disease modeling and drug screening. And I will more focus on this last item. So the disease modeling that we are developing in ISTEM follow the normal paradigm that is usually developed. We are starting with disease-specific human pluripotent stem cell, either from embryonic origin or induced pluripotent stem cell. Then we have the second goal, which is to differentiate efficiently these disease-specific human pluripotent stem cell into cell type of interest. And then a final goal is to use this cell type of interest to either better understand the molecular mechanism involved in this monogenic disease, and consequently to also identify new therapeutic screening. So this is the list of the different pathology that we are working in ISTEM. So you have different pathology concerning neurological disorder. We also have different groups that are interested about neuromuscular disease, such as my group that is interested about amyotrophy, spinal muscular atrophy, and myotonic dystrophy type 1. All the activity that we are developing in ISTEM rely also on a different platform that clearly help us to perform the program of research that we'd like. So we first have a group that is dedicated to cell biology. The focus of this group is to help us to optimize, to standardize the culture of a cell. We also have a platform that is dedicated to the cell production, and it's an automated way. I will go later on this point. We have a platform of screening, a platform of high content screening, and also a platform of omics. For the cell production platform, we have two main equipment. We have a compact T-select automat that help us to make bank of cell. So we can now use this automate to perform either bank of ES or iPS cell, but also some derivative of this pluripotent stem cells such as mesenchymal stem cell, keratinocyte, myoblast, or neural precursor cell. And we also have the second automat that help to the that is with the arrayhead that help to the freezing, to the automating freezing of this cell bank. Concerning the platform of screening and analysis, we also have different equipment. So we have different equipment for the primary screening and the secondary screening, such as the Ben cell, the Bravo, or the BioCell. And we also have a bench of different equipment for analysis, such as the Calario Star for luminescence, QuantStudio for RNA-seq analysis, and different imager as a 6-7 and an automated imager provided by a molecular device. So to go back to my main topic, the different activity that we are developing in my group. So in my group, we are interested to develop new model of neuromuscular disease by using human pluripotent stem cell. And neuromuscular disorder are a group of heterogeneous genetic disease, as you can see on the left side of the graph of the slide. And we are interested about two different diseases, spinal muscular atrophy and myotonic dystrophy. So concerning myotonic dystrophy type 1, it was one of the first goals when we started this program in 2005. It was to bring the proof of concept that we can indeed use human pluripotent stem cell carrying the causal mutation of the genetic disease to identify new molecular mechanism involved in this pathology, but also to identify new therapeutic screening. So very briefly concerning the disease, so myotonic dystrophy type 1 is one of the most prevalent neuropathy in adults. Is characterized by a muscular affection, but it's in fact considered as a multisystemic disorder in which nervous system, ocular system, endocrine system are also affected. And up to now, no curative treatment is available for this disease. So what we have shown in the past, without going too much in detail about the molecular mechanism involved in DM1, it's that by using human ES cells that were obtained during two different pre-implantation genetic diagnoses, that by using this cell and their derivatives, so here was the example of mesenchymal stem cell, we were able to phenocopy the different stigmata normally associated with DM1, has the mutate and the retention, the animal retention of the mutated RNA within the nucleus in association with RNA binding protein called muscleblind 1, which is normally involved in the regulation of alternative splicing, and consequently has observed in primary muscle cell derived from patients. We were also able to detect some splice defect in these derivatives. By using a transcriptomic approach, we have been able to identify new molecular defects associated to DM1 as the one that affect the expression of gene belonging to the SLITRK family, and that we have been able to correlate with a defect in the neuritic outgrowth in neuronal cells derived from this human ES cell. We also have been able to correlate defects in the mTOR expression in association with an autophagy deregulation also observed in DM1 biopsy, as well as the last one was a defect in the expression of a transcription factor called ZNF37A that we showed important in the regulation of myogenesis. So parallel to this mechanistic study, our goal was also to use this new cellular tool to identify new therapeutic strategy. And we have been developing two different approaches. One based on the phenotypic screening, and I won't have enough time today to go too much in detail, but the idea was to identify compounds that were able to disintegrate the mutated RNA aggregation in the nucleus. And we also identified and developed a molecular SMART screening by using scRNA in order to try to rescue the different splice defects that we could observe in a cellular system. And based on this screening, we have been able to identify a scRNA that presents the advantage to be druggable, which leads to the identification of a compound called Metformin that is usually given as an anti-diabetic medication. And we have launched a first clinical trial to evaluate the effect of Metformin on DM1 patients. So this phase 2 clinical trial has been launched in 2014 on 40 different DM1 patients, 20 treated with Metformin and 20 treated with placebo. And now this clinical trial is just ended, and we are still analyzing the different parameters that were evaluated during this 1 year of clinical trials. So a 6-minute walk test, myotome test, muscle RMI, and also other metabolic like ECG, hematology, and alternative splicing. So all these results demonstrated the power that resides in human pluripotent stem cell, either to decipher new molecular mechanisms, but also to identify new therapeutic strategy. However, our results also indicate some limitation, especially when you're interested about neuromuscular disorder. One of the main goals is to have access to the two main cell types that are involved in neuromuscular disease, motoneurons and muscle cells. And I hope to know it was really difficult to generate and to efficiently convert human pluripotent stem cell into motoneuron or muscle cell, and to study the connection between both of these cell types. So a goal now is to try to develop a toolbox for neuromuscular disease. First, by focusing in the understanding and the recapitulation of the in vitro generation of cellular compound involved in this neuromuscular system, and I will present you the work that we are doing concerning motoneurons. Also to improve our knowledge about the communication and the muscle cell by developing new cellular tools to better understand how motoneurons connect to muscle cell, and of course to apply this different toolbox for neuromuscular disease, for study ranging from mechanistic analysis to therapeutic screening. So concerning our first goal, most of the protocols that were described in the literature concerning the conversion of human pluripotent stem cell into motoneurons were based on a very long protocol, more than 1 month, and not really efficient, as only 25% of motoneurons were generated from human pluripotent stem cell. And all these protocols were based on the use of retinoic acid to caudalize and the use of sonic etch-up to ventralize the different progenitors. So as these protocols were not efficient, we were working with a lot of, not pure cell culture, with a lot of contaminated cells and very few cell type of interest. So we decided to try to combine what we knew to do well in the lab, screening, with a developmental question. Can we try to identify and to improve this protocol in order to generate a pure population of motoneurons? So for that, we developed a strategy in which we play on 2 different factors, the concentration of different morphogenes, but also the timing of action of this morphogene. And by using high content screening platform, we have been able to generate a protocol that allows us to obtain only in 16 days more than 80% of spinal motoneurons, as depicted here by the presence of HP9 expression markers here with the red dots. The next question following this result was what type of spinal motoneurons are we able to generate? Indeed, there are several types of spinal motoneurons along the spinal cord that are connected to different muscles. So in function of the pathology that you are interested, it's really important to generate the right type of spinal motoneurons. So we used recently the OX gene code that is able to distinguish between cervical, brachial, thoracic, and lumbar motoneuron. And we've been able to show that the protocol that we have recently developed was producing cervical, early brachial motoneurons. So we are now trying to develop new protocol of differentiation to generate more posteriorized spinal motoneuron, because our goal is, for example, to generate lumbar motoneurons that normally innervate the leg and that we know are affected in neuromuscular disease. So we are again playing with different morphogene and also concentration and time of action of this morphogene. And by looking at again at the expression of the different OX gene, we've been able to identify a condition in which we are able to generate thoracic motoneurons and also lumbar motoneurons. So now we are now working on the mechanism to try to understand better the mechanism by which developmental cues control motoneuron velocity. And of course, our goal now is to use these protocols and to apply them to different neuromuscular disease. And one of the neuromuscular disease in which we know that spinal cords are directly affected and degenerate, it's spinal muscular atrophy. which is a rare disease, and that is, again, clearly defined by the specific degeneration of spinal motor neurons. Despite the fact that different pieces of treatment have been identified recently and are actually under a clinical evaluation, it's still not clear why SMN, which is due to mutation in the SMN1 gene, leads to the specific death of subtype of spinal motor neurons. So different reports have shown that the possibility to use human iPS technology to reproduce the cell death or at least inefficient production. But the distinction between cell death or inefficient production of motor neuron were not very clear in this publication. So by using a protocol, we have been able to show that SMN reduction is not does not lead to a defect in the generation of motor neurons, but specifically to a decreased survival of motor neurons. So if we look at different time points after motor neuron birth, we can see that we have a specific decrease in the motor neuron survival at later time points. So now our objective is to try to better understand with the recent development that we have made about the generation of subtype of motor neuron, so to better understand why SMN reduction is associated with a subtype specificity. We are also interested about the neuromuscular junction defect described in SMA. We know that abnormal structural defect has been described at the neuromuscular junction in association with SMA. So the idea is to try to reproduce these defects. And for that, we are recently developed a program in collaboration with different partners, like the Society Sci2, also Dr. Leguet from Paris Descartes, and Dr. Schaeffer at INMG Lyon, to try to develop an in vitro model combining motor neuron derived from iPSC with human primary myotubes that are maintained in micropattern myoplate in order to develop a new human neuromuscular junction system. And the idea is to apply this new cellular tool to different pathology that we know affect the presynaptic part, like SMA, but also the synaptic part, like CMS, and also the postsynaptic part, like myotonic dystrophy type 1. And the idea is to use this tool both to identify new biomarkers specifically affected under neuromuscular junction, and also to perform drug screening. So the advantage of the micropattern plate developed by Sci2 is that they're already in 96-well format. Sci2 has been described that this plate and this micropattern, the shape of this micropattern, induce a myogenic maturation, and that it's compatible with screening and molecular studies. So here is some example of the culture that we have with muscle cells in red, motor neurons in blue, and in red you can see the neuromuscular junction. And if we are looking in 3D, you can clearly see, so here the motor neurons, it's in yellow, muscle in gray, and in red you have the aggregation of the receptor to acetylcholine, which is a sign of synaptic maturation, and you can clearly see the right opposition between both structures. And here the quantification is what here is the automated, on the left part of the slide, it's the automated quantification of the number of aggregation of acetylcholine receptor that we can detect after incubation with alpha beta in the culture, between culture without motor neurons and culture with motor neurons. So now the idea is to apply this cellular tool to different pathologies, to these different disease, and to try to observe exactly what happened at the neuromuscular junction. So I would like first to thank all the people, past or not, that participated to this work. So mainly Stéphane Nedelec that have worked a lot and helped us a lot in concerning the protocol of differentiation of human pluripotent stem cell into motoneurons. So now Stéphane has created his own group and is working at Institut Ferrat-Moulin in Paris, but we are still really connected concerning all the program about the subtype of motoneurons. And just to finish, I would like to spend a couple of minutes about the French Society of Stem Cell Research that has just been launched recently in France. So we realized that in France, the community of stem cell was not well organized and was really sparse across the country. So we decided to gather all expertise, and this is the reason why the French Society of Stem Cell Research has been launched. So please visit our website. If you have any question, you can contact us to this web email. So I'm the president of this French Society, and this is the French vice president, Sharagen Tajbak and John Devos from Montpellier. And this is all the members that belong to the scientific board of this society. And just save the date, so next 22nd of November, we will organize a first meeting of the society that will be held at Institut Pasteur in Paris. As main speaker, we have Ludovic Vallier, Frédéric Blancpain, Emmanuel Hayal-Sebag, and Lars Denbemets. But you can still apply with an abstract to participate to this great day. I'm hoping to see you there. Thank you again for your attention. I would like to thank Thermo Fisher and the 24 Hours of Stem Cell for giving me the opportunity to present you these different results. Thank you again.